Jai Shri Gurudev, with the divine blessings of His Holiness, Bhairavaika Jagadguru, Dr. Shri 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 Balagangadharanatha Mahaswamiji, and bowing to the lotus feet of Paramapujya Jagadguru, Dr. Shri 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 Nirmalandanatha Mahaswamiji. Respected Principal Ma'am, our resource person, HODs, colleagues, and dear students, welcome you all for this national webinar on Bioactive Compounds from Natural Sources, Identification and Applications. Now I request Dr. Nagaprashant sir to inaugural speech.
Good morning, one and all. Greetings from Sri Adichunchanagiri College of Pharmacy, Faculty of Pharmacy, Sri Adichunchanagiri University. I would like to welcome you all for this webinar, which is being telecasted in YouTube Live. The title of this webinar is Bioactive Compounds from Natural Sources, Identification and Applications. So, respected dignitaries like our respected speaker, Dr. Anjana, our respected principal, Dr. Bharati DR, our HOD, Dr. T.Y. Pasha, my colleagues, HODs, and my dear all participants, welcome to this webinar. The topic of this webinar is the need of the hour. Starting from COVID, we have all started searching for phyto alternatives and started altering these phytomolecules to enhance the potency of these molecules. So, identifying this requirement, we have organized this webinar on this topic. And for this particular webinar, we have received 460 registrations from 208 colleges across India. And not only India, we have participants from 3 other countries like Saudi Arabia, Qatar and UK, United Kingdom. This itself proves that the potency of the chosen topic. So, with these initial remarks, I would like to hand over the session to Manoj.
Now I request our beloved Principal Ma'am for initial remarks.
Jai Shri Gurudev. My humble pranamas to Paramapujya Jagadguru Shri 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 Dr. Balagangadharanatha Mahaswamiji and Paramapujya Jagadguru Shri 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 Dr. Nirmalandanatha Mahaswamiji. Myself, Dr. Bharti Dia, Principal, Dean, Faculty of Pharmacy, Sri Adichunchanagiri College of Pharmacy. I am very proud to introduce you about Sri Adichunchanagiri College of Pharmacy, which belongs to Adichunchanagiri University. Adichunchanagiri University is a 5-year young university which is NAC A+ accredited. When it comes to Faculty of Pharmacy, Sri Adichunchanagiri College of Pharmacy, it has 40 years legacy and we have NIRF ranking continuously since 5 years, and Sri Adichunchanagiri College of Pharmacy is NBA accredited, and it is it has a very innovative IIC cell as well as Pharmacy College is NBA accredited, and our quality initiatives are enormous. And I really thank our speaker, beloved speaker madam, for accepting and for giving us time to have this wonderful webinar. Thank you so much, and I request Manoj to continue.
Thank you, ma'am, for your remarks. Okay. Now, it's an honor to introduce our esteemed speaker, Dr. Anjana. She currently working as a professor in NIMS Institute of Pharmacy, NIMS University, Jaipur, with experience of 18 years in both teaching and research. She done her UG in a JSS College of Pharmacy, and she completed her PG and doctoral studies from Andhra University. She has received numerous awards like KK Acharyaji Memorial Award in 2002, RSVL Gold Medalist, stood All India First Ranker in Diploma in Pharmacy in 2002. She has also received Best Teacher Award 3 times in her career, and she published 96 publications, 11 books, 5 book chapters. She also received various grants from different organizations, AICTE, DSD, SCRB, CICS, and she patented 6 patents. She granted 6 patents, and she delivered the guest lectures in various organizations. And she also worked as a IQAC coordinator and president of Nirmala Innovation Incubation Center. Madam, over to you, madam.
Thank you, sir. Thank you, Manu, sir. And first of all, beginning this webinar, I would like to thank the faculty of pharmacy, Sri Adichunchanagiri College of Pharmacy, and Dr. Bharati ma'am, and thank you, ma'am. And also, I would like to thank the HOD of the chemistry department, that is Dr. Pasha sir, and I would like to thank the person who has taken all the pain to arrange this webinar and all these things, Dr. Prashant sir, and for reading my resume, Dr. Manoj sir. Thank you very much. And it's my honor to give this guest lecture with your Adichunchanagiri College of Pharmacy. Thank you very much. Sir. So let me start my presentation, if you, with all your permission.
Yes. Proceed, ma'am.
Okay. You can see this one, sir.
Yes, ma'am. Yes, ma'am.
Lines are visible, huh? It's moving.
Yes, ma'am. We can see.
Yeah, today's topic is bioactive compounds from natural sources. As we know that during the COVID time, many of us, we have faced the situations where the people are in line with the hospital, and also we have seen that there is a huge demand has been started from the time of COVID, and also from that time also, before that also, we are having a lot of Ayurvedic products or herbal products are in process, but in India, the awareness has been started from the COVID time onwards. And still from that time also, every year we are facing different kind of virus situations. For all these virus situations or any type of treatment we require, we can able to get to know from the natural sources. If we can able to use these bioactive compounds, it will be a safe and effective manner. So before starting the information related to this webinar, I just like to say the objectives of the webinar. Right. So this webinar will be educating you, providing the foundational understanding of what bioactive compounds are, what are these bioactive compounds, where they came from, and their chemical characters you'll get to know. And there we can able to already discuss about this education. Apart from this, we'll be discussing about the what are the techniques used for the extraction process of the bioactive compounds, and also nowadays we are finding the various analytical techniques to identify, to analyze these compounds. So these techniques also we are going to discuss. And apart from that, we'll get to know that what are the various applications where we can able to use these bioactive compounds, like in pharmaceuticals, nutraceuticals, cosmetics, agriculture sector, in all the other things we'll be getting to know. So the main objectives of this webinar is this: to educate you, to determine the techniques which are used for extraction purpose, to give you the information on the application orientation. So first we'll go with the first topic, that is education about the bioactive compounds. So coming to the definition of the bioactive compounds, bioactive compounds are naturally occurring chemical substances found in plants, animals, microorganisms, and that exert a biological effect on living organisms. So it doesn't mean like bioactive compounds are nothing like any some superstitial or any other compounds. These are the generally occurring which will be finding in and around our cells, but identifying them and separating that compound, using that compound in a required manner is also very important aspect to know, to have the biological effect or to treat any kind of disease. So these bioactive compounds will be finding in many of the products like plant sources, animal and all. So these bioactive compounds are, what are these chemically? So here the type of represents the bioactive compounds are nothing any other new compounds. Whatever we are getting from the natural sources in different forms, like in the minerals, in the form of minerals, we'll be obtaining in the form of phenolic compounds. We will be obtaining amino acids, proteins, pigments form. These are available in the plants are fruits, vegetables, and all, vitamins, polysaccharides, and lipids. So we'll be finding these bioactive compounds in the various plant products, in the various organs of the plants, in these various forms, like mineral form, phenolic compounds. Most of the plants which are having the antioxidants, that is because of the phenolic compounds only. And we'll be finding the amino acids which are required for the essential for the body and as well as non-essential also. And the proteins which are majorly we see that which are having the many types of functioning, like protecting the cell and the various organisms, and also helping in the transportation process. Various applications we could able to find these proteins are involved. And pigments, whatever the coloring compounds of the plants which we are finding, that is the color of the fruits or vegetables, plants will be finding these pigments. So these vitamins are also helpful for absorption purpose. So we'll be finding the presence of these bioactive compounds in the form of polysaccharides also, and as well as in the lipids form. So I'll show you this picture of this biochemical composition, like the pigments. It is available in the form of various color pigments in the plants, in the form of the fruits or vegetables we'll be finding, or from the tubers we'll be finding. So these are like carotenoids, which basically we'll be finding in the carrots, beetroot, in all this. So this is a compound which can be isolated from the carrots and beetroot, carotenoids. Chlorophyll pigments, so which we can able to find in the all the leaf and aerial parts of the plants. The chlorophyll is the pigment which is present, and phycoerythrin, which is a well more one more compound which is mostly used in many types of cosmetics. The lipids, it is also these bioactive compounds are also available in the form of the lipids. Majorly, we'll be finding in the essential polyunsaturated fatty acids. So nowadays, we might have seen during the COVID time, we have used many types of vitamin D capsules. Even now, people are using this vitamin D, which is major source. Then proteins, the synthesis of essential amino acids will be carried out from the proteins which are obtained from the plants only. And we could able to know that all the fruits and vegetables are having the multiple vitamins, like fat soluble and water soluble vitamins. So these vitamins also we could able to isolate, and that can be given directly. So we could able to find many types of minerals like calcium, zinc, copper, iron, all these type of minerals, like in the form of macro and micronutrient form, will be finding in the plant products. So this is also available in the form of polysaccharides. So these polysaccharides, they are having the majorly related to the action, which is like as an excipient form instead of therapeutic agent. So these pigments, lipids, proteins, and all are having the therapeutic action, vitamins, but the polysaccharides are majorly having the excipient property. So hence these polysaccharides are used as a gelling agents, as a thickeners in many of our food products, food industry we are using, right? So this composition, based on the composition, we could able to isolate the particular bioactive compound, and then that can be used directly in the industry. So nowadays, so we could able to find there is a various types of jam. Like I cannot able to say the particular company, but nowadays we are finding that fruit jam we are getting at less cost. That main reason of that less cost is because of this polysaccharide that is called as pectin, especially that is been isolated from the plant part, especially from the rinds of citrus fruits, and that is been mixed in the jam preparation. So adding coloring and flavoring agents. So by means of this only we could able to obtain these jams at low cost in the market. Next, we could also find the other bioactive substances which are majorly playing an antioxidant property like polyphenols and fluorotannins. So these are the major compounds which are having the antioxidant and protecting the cell from the microbial attack. The next one is the relevance of bioactive compounds. How these bioactive compounds are useful. Okay. So these effects can range from promoting health and well-being to provide therapeutic benefits against various diseases. So we could also find that these bioactive compounds will be showing a very good activity than compared to the other products. The study and application of the bioactive compounds have gained significant attention in recent years due to their potential to provide sustainable and effective solution in medicine, nutrition, agriculture, and more. So we could able to find that we are using these bioactive compounds not only in the medicinal purpose. We are finding in the food industry the nutritional aspects to increase the nutritional aspects. Agricultural sector, we are finding this usage. So the main traditional uses of these are like we could able to find like how we can able to identify that particular plant is having the bioactive compound or particular animal or microbial source is having the bioactive compound. So this is like based on the traditional uses like folklore uses. Many of the times we'll be finding that the elders who are this plant is very auspicious, it is having the very good action like that. So that property is majorly because of the this traditional uses only we could able to identify, and then we can able to isolate the particular compound. So the process of this identification is carried out from the medicinal plants. Like many modern medicines have roots in traditional uses of plants. For example, the use of willow bark for pain relief, like this is the bark which is used for pain relief from the olden days, but later on they have identified the aspirin compound from this willow bark. Willow bark, this has been separated, and that means nowadays we are finding that aspirin in the market also in the form of the tablet. So, but for the first thing, it was identified from the willow bark. Later on, the particular aspirin has been characterized, structural identification, and synthesized in the lab. Based on that only, we could able to obtain this aspirin tablet in the market now. Next one is the based on the herbal remedies. So like we can able to select the isolation of the compounds by the herbal remedies also. Like traditional herbal remedies often involve complex mixtures of plant extracts. Many of the times, whatever the herbal remedies which we are using, they will be having the not only a single plant. Many of the times, we'll be using the mixture of different plant extracts. So due to this mixture of plant extracts, we could able to find the synergistic effect. So we can able to find the potentiating effect, like by means of using a single plant extract, we may find the effect, the treatment can be like within 10 to 15 days, but by means of using the mixture, we will be able to find within 2 days of treatment can be obtained. So understanding these traditional preparations can inform modern extraction and formulation methods. So in the older days, we used to have the extraction processes only through succinate or by means of the maceration process. But nowadays, we are finding the many types of extraction methods which can be useful for various types of extraction process. And these extraction methods help us to get the pure compound, even more best type of without having any type of mixture, we can able to isolate these bioactive compounds. Next, from the cultural practices also, we can able to isolate these bioactive compounds. Like traditionally, we'll be having every geographical area will be having one tree or one plant will be considered as auspicious. Like for example, Tataki Pushpa, which is in the Tandas, these people will be believing that this plant is a goddess plant because this Dataki Pushpa has got the many of the antioxidant properties. So, and that is being used in many of the Ayurvedic formulations also for the nervine stimulation and all. So, like that, every geographical area will be having the some plants which are having the traditional usage. So, these cultural practices also help us to identify the bioactive compounds which are present in this particular plants or trees or which are obtained from the animal source also. So the traditional uses are often intertwined with the cultural practices and beliefs which can influence the selection of plants and preparation methods. By means of using these traditional practices, we can select the plants and we can able to make the different preparation methods. The techniques of extraction and isolation. So in the olden days, as I said, like before we are having only the two methods. Majorly, we'll be doing the extraction, that is direct boiling or by means of oxidation. But nowadays, we are getting the various types of equipments and different types of technologies also for isolation purpose and extraction purpose. So present current trends are green extraction methods. As we know that during the extraction process, we'll be using the both polar and non-polar solvents. So by means of this extraction, green extraction methods, whatever the solvent we are going to use for the extraction, it will be environmental friendly. It cannot be damaging any of the species and it is eco-friendly also. So majorly we'll be using the solvents like water and mixture of ethanol and water, or we'll be using the liquid gases are used, so which are environmental friendly. So they'll not be harming the species, environmental species, as well as the persons who are involved in the extraction method. So this will be like giving you the 100% extraction can be obtained and also we can get the pure compounds. Right. The next one is the omics approaches. So here, by means of the technology like genomics, metabolomics, proteomics, so which are used for bioactive discovery. So by means of the identifying the genetic structure, and we can able to find out that which is the gene responsible for the bioactive compound production in a particular natural species. We are going to identify the gene can be synthesized in the artificial manner, or we can able to isolate that gene and can able to show the action. So nowadays, for example, we are finding the different types of plants where without seedless varieties like hybrid varieties we are finding. So all these are because of this omics approaches only. Next is a nanotechnology. By means of using the nanoparticles, we can able to isolate the compounds. So delivery systems like nanoparticles and phytoforms for improved bioavailability. Like based on this nano particular size of the solvents, we can able to find the even more better extraction process than comparing with the traditional approaches. So everybody knows the traditional approaches. That's why I'm not going in detail with the traditional approaches. Just I'm concentrating on the current trends only. The next is a synergistic combinations. Like we'll be using the different types of combinations of the compounds. So thereby we could able to find the better therapeutic action. Combining the bioactive compounds for enhanced efficacy, we are going to use these synergistic compounds combinations. So this is the process how we do the extraction process. Extraction, that is like from we are going to identify the plant kingdom or animal kingdom or from microbial kingdom based on the traditional approaches or any other methods like folklore approaches and all. Then we are going to select the particular medicinal plant material, which part of the plant we need to take. And from that, we are going to prepare the extract, that is the plant extract. We are going to prepare by means of the traditional methods or current trends. And after that, the extract is having the therapeutic action or not. That we are going to identify by means of the biological activity screening. So by means of this biological activity screening, if the particular extract is showing the better action, then we'll be going for further isolation of the compound. If it is not showing the activity, then we can able to go with the synergistic combinations or any other methods. Thereby, we could able to isolate the specific bioactive compound. So after identifying after its potency in the biological activity, then we will be using the analytical techniques. So these analytical techniques will help us to separate the particular compound and to identify its purity also. And thereby we are going to separate this bioactive compound, and then later on we'll be going with the structural elucidation, structural identification and structural elucidation will be carrying out. And after that we'll be just finding the in detail process like how we could able to synthesize this molecule in the laboratory. In that way only we could able to find many of the compounds in the market that we'll discuss in the next slides. So various extraction methods which are available now, apart from the current trends, majorly used. One is the recently available one is the ultrasound assisted extraction. So by means of using the ultrasound waves, we are going to apply the extraction process, thereby there will be complete extraction will be carried out. Nowadays, this is the one more important approach where we are doing the extraction process. Next is the accelerated solvent extraction. So we are going to enhance the temperature of the solvent, and then that particular solvent, like hot solvent, is used for the extraction process. So thereby we'll be finding the a better complete extraction will be there. Next is the pulsed electric field assisted extraction. Like here we are going to during the extraction process, obviously we need to use the thermal energy. So here just like the energy is used, the electric field is used like pulsed, like slowly increasing and decreasing, increasing and decreasing. In that way we are going to do the extraction process, thereby we could able to find the active constituents will not be damaged in this manner. Pulsed electric field assisted extraction. Next is the supercritical fluid extraction. This is the somewhat cost of extraction is more here, and the supercritical fluid extraction, because here we are going to use the gaseous method. It is also considered as a gaseous method where we are going to convert the gas into the liquid form by means of applying the pressure, and then that liquid is passed on to the extracted material. Thereby we could able to find the extraction process. So this supercritical fluid extraction, it is majorly used nowadays for isolation of the perfumes or volatile oils from the medicinal plants. Next is the enzyme assisted extraction. That is, we are going to use the enzymes. So by means of these enzymes, the breakdown process of the molecules which are present in the cell will take place. So thereby you could able to find that the particular material will be easily coming out of the cell. So this will be like easy process of getting the extraction. Next is the green solvent extraction, which already we have discussed that we are going to use the solvents which are eco-friendly to the environment, and it will not be damaging the any type of environmental species like water, and mixture of water along with the ethanol or methanol will be using this extraction process. Next is the solvent extraction method. So by means of using the different solvents in combination, we'll be extracting the material. The next one is the microwave assisted extraction. So this here we are using the microwaves for extracting the plant material. So thereby, based on the frequency of the waves, we can able to find the extraction process. These are the various extraction techniques which are available recently in isolation of the extraction of the bioactive compounds. So here is the neuronal graph which is given for the isolation and purification of the compound. So we need to like a bit zoom in is required here. So you can find here there is a in the using the ultrasound waves. And here will be like solvent penetrates and releases the extract. The compounds can be released into the compound. Next is the by means of various methods like separation process, like fractional distillation. Fractional distillation is the one where we can able to separate the two similar type of compounds which are having the structural similarity or molecular similarity. Those type of compounds can be separated by means of the fractional distillation, and which are having the similar boiling point also can be used by means of this method. So here we'll be using the heating and collecting the fractions. By increasing the temperature at various temperatures, we are going to collect the fractions. And here the basic principle is separates based on the boiling point. This fractional distillation is going to work. Okay, so here we got to know that extraction process and then separation process. And one more separation process. The fractional distillation can be used to those compounds which are thermal resistant or which are having the similar type of boiling point. Those type of things can be separated by fractional distillation. And we'll be using the chromatographic techniques. So here in this chromatographic techniques, based on the particle size, based on the like absorption coefficient, and based on the partition coefficient, here the separation will be carried out. So here in the chromatographic technique, commonly generally used technique is the column chromatography, which is a traditional approach for separation of the compounds. We'll be running the column by means of the extract with the permutations and combinations of the various solvents, and thereby we could able to find separation of the compounds. So here the principle involved in this column chromatography is differential adsorption process, and based on the process of elution with the solvents we can able to separate the compounds. Next we could able to find that there is a HPLC, high performance liquid chromatography is the equipment where we could able to separate the compound, but in column chromatography we could able to obtain the compound at large quantity, and we can also keep the extract in large quantity for the separation. But in HPLC it is only for the qualitative purpose. Column chromatography is for quantitative purpose. You can able to get the quantity of the compound also in the large quantity in case of column. But in case of HPLC, the amount we are going to obtain is very less, and it is like in the minute concentration, and we could able to find that whether it is a quality can be checked by means of the HPLC. So here the basic principle is the interaction with the stationary and mobile phases, and the process is injection. We are going to inject the extract into the HPLC column, thereby separation will take place based on the adsorption, and we can able to use the any kind of detector for detecting the compound. So here one more method which is traditionally used for separation process is TLC. So before going for the column, we'll be identifying the compounds, the type of compounds present in that, and how many number of compounds are present in the particular extract, which is carried out by means of the thin layer chromatography, which is also called as TLC. Based on the differential affinity, the principle involved in the TLC is the differential affinity, and based on the developing in the solvent system used and type of the detecting agent we are using, we could able to find out that number of compounds present in that, and also we could able to find the separation of the compounds. So these TLC and the column are the traditional techniques. Next, coming to the purification, like we have done the extraction, we have done the isolation, and then we have to find out whether the obtained compound is pure. If it is pure, that is fine. If it is not pure, it is having the mixture, then we need to apply the some of the purifying techniques. So here are the some of the purifying techniques, methods for purifying the bioactive compounds. One is the centrifugation method. By means of using the centrifuge apparatus, but like the process is spinning to settle the compounds based on the density, the compounds will be settled here, the centrifugation method. And one more method which is used mostly is the recrystallization process. By means like if the compound is there but it is not separated in the solid form, then we are going to use a different type of solvents to make the compound immiscible with the solvent, or there will be crystallization of the compound from the solvent will be taken place by means of the recrystallization process. So the principle is solubility in a solvent. Based on this, we can able to dissolve and cool the crystallization. Next is the liquid-liquid extraction method, which is used where we can able to use the partitioning of the compound between the two liquids by means of our normal separating funnel. We can able to carry out in the lab phase this separation, that is liquid-liquid extraction. Majorly for the volatile oil compounds or resinous compounds, this liquid-liquid extraction is best. Next one is the preparative chromatography, where we are going to use this chromatographic technique for separation of the compound, which are of having the similar molecular weight or similar uh, retention time. So this can be done for large scale purification, and it is also used for loading, collecting the fraction we are going to carry out by means of the preparative chromatography. So here we are doing the one more technique nowadays, which is used is the dialysis process. Nowadays, whatever this RO system is working, or your kidney dialysis process is working with this process only, dialysis. That is like here we are going to separate using a semi-permeable membrane the separation of the compounds, and diffusion will be occurring through the membrane. The compounds will be diffused, and thereby we are going to separate. This dialysis process is mostly used if the compound is like slightly formed in the solid form but is mixed with the solvent. Then this dialysis process will be helping us. Next is the crystallization. So most of the times we'll be finding the separation of the compound, but the separated compound will be having the some adulterants or some mixtures will be there. So to separate these mixtures, we'll be using the process known as crystallization. Next, in this crystallization, we'll be using the forming the crystals from a solution. Basic principle involved here, and we'll be inducing the crystallization with the help of the various solvents. Next one is the membrane filtration. So by means of using the size-based separation and passing the mixture through the membrane, we are going to separate these compounds using the membrane filtration method. So this is a neuronal network through which we can able to separate the compounds. So when we are separating, so after that, what next? We have extracted, done the separation, purification has been carried out. We could able to find the compound. So what is the applications? So let me zoom it. What are the applications of these compounds? So we could able to find the diverse applications of these bioactive compounds. One is the pharmaceuticals. In the drug discovery and development, we'll be finding. So in the drug discovery, they are majorly used as a therapeutic agents. So the bioactive compound serves as a basis for many drugs used to treat disease such as cancer, cardiovascular disease, infections, and neurological disorders. For example, paclitaxel, which is a compound isolated from Taxol from the Pacific yew tree, is used in the cancer therapy. So this paclitaxel is the one which is identified for cancer treatment. This is obtained from the bark of Taxol, which is like a yew tree. It's called as yew tree, Pacific yew tree. And the lead compounds, like natural bioactive compounds, often serve as lead compound for drug development process, providing a structural base for synthetic modification to enhance efficacy and reduce toxicity. So these lead compounds are nothing but the, like we have seen in the earlier slide, that aspirin is the compound molecule which has been produced from willow bark. So this is considered as the one of the example for the lead compound. First, we are going to identify the bioactive compound which is having the activity, and then later it is synthesized in the lab for easy process of production and then better efficacy. The other main actions are anti-inflammatory and analgesic. So like in the pain management, we are finding the usage like salicin from willow bark, which lead to the development of aspirin, are used for their analgesic and anti-inflammatory property. And chronic inflammation, like bioactive compounds such as curcumin from turmeric are being explored in the role of managing chronic inflammatory diseases. So we could able to find the curcumin from the turmeric is found to be a good anti-inflammatory agent in many types of inflammatory diseases. And not only that, we could able to find the usage of the bioactive compounds which are obtained from the natural sources, like in the form of polysaccharides or saponins, phenolics, organic sulfur, and we could able to find these uses in various treatments like anti-diabetic activity, anti-cancer activity, cardiovascular protection, neuroprotection, hepato and renal protection, respiratory system protection, like in the asthma treatment and all, and in the digestive system protection, like gastric ulcers and all these cases, in the reproductive system protection, in the immune modulation, like ashwagandha is one of the more methanolites which have been isolated from the ashwagandha are found to be immunomodulation, antioxidant property, so which is found to have the good activity, antimicrobial activity, anti-inflammatory activity, and we could able to find nowadays anti-obesity. So for reducing the obesity, also we could able to find the many types of uses from the bioactive compounds. Not only in the pharmaceuticals, in the therapeutics, we could also find the applications of these bioactive compounds in the nutraceuticals and functional foods in the form of vitamins and minerals. We are finding the usage of these bioactive compounds. Like nowadays, we find, for example, this baby food products which are we are getting. So these baby food products which are available in the market, it contains the isolated bioactive compounds. Hence, we could able to find the activities even more good than compared with the traditional food methods. Next, herbal supplements like citrus fruits, ginseng, green tea, which are very well known for the herbal supplements. As we see, antioxidant property, obesity reduction will be carried out by means of these herbal supplements. So these are the like many types of applications which we could able to find the citrus fruits, especially like we could able to find the bioactive compound separation, extraction of essential oils. These are used in the food industry and food additives, pharmaceuticals and cosmetics. It is used biodegradable packing material form also. These citrus fruits are being used. That is a pectin which has been isolated is also going to replace the gelatin. So like helping us to have the better and therapeutic action is also good. Next, we can also find that the nutraceuticals, so that is enhanced nutrition foods which are fortified with bioactive compounds such as omega-3 fatty acids from fish oil or antioxidant from berries offer additional health benefits beyond basic nutrition. Like normal food methods, we can able to follow, but these bioactive compounds, isolated food compounds, are going to provide you a good amount of energy. And we could also find these bioactive compounds in the form of probiotics and prebiotics, so which are going to help us in fermented food form, which will be helping us for gut health. These bioactive compounds are involved in the management of chronic diseases also. Like one of the good compound is the resveratrol, which is isolated from the grapes, and flavonoids from the fruits. You could also find that they are even more useful in the chronic diseases like heart diseases and diabetes also. So we could able to find these are the diverse applications in the nutraceuticals. And apart from that, these bioactive compounds are used in the cosmetic industry also, like anti-aging products, like aloe vera gel, which is used as an anti-aging, fragrance and aromatherapy, which we are using for the aroma treatment for reducing the tension and stress. The hair care products for improving the structure and shining property, and skin brightening and whitening process, natural pigments and colorants, and in the form of photoprotective and also in the moisturizer form. These are used in the cosmetic industry. So here we see some of the examples of the bioactive compounds and their applications. So the first one is the curcumin, which is a well-known and everybody knows that it is obtained from the curcuma longa, and it is a polyphenol with anti-inflammatory, antioxidant, and anti-cancer property, which is used in the arthritis treatment, cancer treatment, oxidative stress. And from recent research which has been carried out on the patient, it has been shown that this curcumin at a dose of 1 mg dose has found to reduce the side effects shown by means of the radiation therapy in the cancer patients. So this is one of the current research which has been shown for the curcumin at a dose of 1 mg is found to be a good therapeutic action for reducing these side effects of the radiation. So this can be done by means of solvent extraction. Various methods are available for extracting this curcumin. One is the solvent extraction, that is using the solvents like ethanol, acetone or methanol we are going to extract. Then by means of using the rhizomes extracted using organic solvents and concentrated, and finally it is purified by using the column chromatography. Next, supercritical carbon dioxide extraction by means of using the carbon dioxide in the liquid form we are going to use. And next is the microwave assisted extraction, which improves the extraction and also the time will be reduced by means of using the microwave energy. Next, there is a compound known as resveratrol, which is having a multiple benefits, which is isolated from the grapes, berries, and peanuts. It is having the antioxidant property and anti-aging property. Apart from this, it is used in the formulations like for cardiovascular diseases, neuroprotection, and is also used as a nutraceutical, as an anti-aging dietary supplement, used in the cosmetics for UV protection purpose. This is extracted by means of the solvents of ethanol and water in the ratio of 70:30, and then the process is like grapes are crushed and extracted with the solvent, and it is purified with the liquid extraction or chromatographic method. Apart from this, we'll be using ultrasound assisted extraction, and supercritical fluid extraction can be used for the isolation of the resveratrol from the grapes. Next is the quercetin, which is a well-known compound which is isolated from the onion peels, especially apples and berries. These three are the major sources, and we could able to find the anti-inflammatory, antiviral, and antioxidant property of this quercetin, and is also helpful for supports the immune functioning and reduces the allergy symptoms. It is also used as a nutrient supplements in the heart health protection and immune support, and is also used as a natural additive for antioxidant purpose. It is extracted by means of the acid hydrolysis process, where onion peels are treated with dilute HCl to release the quercetin from its glycosides, and thereby it is purified using the liquid extraction or recrystallization process. It is also isolated extracted by means of the microwave assisted extraction, and enzyme assisted extraction methods are used. Next, omega-3 fatty acid, which is available in the market in the form of a soft gelatin capsules. And the major source of this omega-3 fatty acids is fish oil, and those for vegetable source, that is flax seeds and algae. These two are the vegetable sources which will be supplying the omega-3 fatty acids. As we know that it is majorly used for anti-inflammatory and cardioprotective action, and it is also used in the capsules like for managing the hypertriglyceridemia and support the brain health. In the nutraceutical purpose, it is also used in the fortified foods like milk and yogurt, which is a rich source, and it is also used, and it is also used in the foods and beverages preparation. The main process of preparation of this omega-3 fatty acids is cold pressing method. We'll be just taking the livers and then from the flaxseed by means of extracting the oil, and solvent-free extraction, that is like we are going to use the carbon dioxide extraction to avoid the any type of chemicals that are present, and we'll be using the enzymatic hydrolysis process also. So here the enzymatic hydrolysis is the one which is helpful for carrying out the extraction of the omega-3 fatty acids and from the lipid mixtures. Next is epigalactocatechin gallate, which is major constituent obtained from the green tea, Camellia sinensis. So it is a good potent antioxidant and is having the anti-cancer properties. So nowadays we see that many of the people will be having the green tea, but the isolation of the compound will be making you to use the precise way and have the good therapeutic action without having any other side effects. So the main uses are in the weight management and cancer prevention. So in the cosmetic industry, it is used as an anti-aging and acne control, and it is also used as a nutraceutical. The main process of extraction is hot water extraction. Thus, it is a water soluble compound, so hence easily we can able to separate by eco-friendly method. Organ solvent extraction is also used, like thereby by using the ethanol or methanol, and then later we are going to evaporate to obtain the extract. And supercritical fluid extraction, that is carbon dioxide extraction, also can be carried out. And chromatographic separations are used like HPLC method to purify the epigalactin. Next is the lycopene. Lycopene is the compound which is majorly identified in tomatoes, watermelon, and pink guava. So wherever the fruits or vegetables which are having the red color, they are having the rich of this lycopene. The main major character of this lycopene is it is a carotenoid. Carotenoids are present in all the red color compounds. So it is having the antioxidant property and photoprotective property. The major uses are used to prevent prostate cancer and support skin health. It's also used in sunscreens, anti-aging cream, which is used as a natural colorant and antioxidant in processed foods. So nowadays we are finding that many of these restaurants and all, they are using this lycopene as a coloring agent. Next, organ solvent extraction is a process which is used for isolation of the uh, lycopene, like hexane, ethanol, and acetate, which is a non-polar compounds. Then tomato paste or peels are processed to extract the lycopene, followed by concentration. This is the method which is used for organic solvent extraction. Next is ultrasound assisted extraction is the method which is used to reduce the solvent usage, because as we are using the non-polar solvents that may be toxic also to the living species. So by means of using the ultrasound assisted extraction is the best one for isolating the lycopene. Next is the supercritical carbon dioxide extraction is also used, where we can able to avoid the non-polar compounds. Next is the berberine, is the compound which is obtained from berberry species. It is an alkaloid with antimicrobial, hypoglycemic, and anti-inflammatory property. It is majorly used in the diabetes management, gut health protection, including in supplementary for metabolic syndrome like diabetes and weight reduction, and is also used in the veterinary compounds for controlling the infection in the animals. It is majorly extracted by means of the acid alkaline extraction method. So by means of using the acid solvent, we are going to extract the berberine, and then it is neutralized by using the alkali. So thereby the berberine compound is precipitated out. And we can also use the other methods like salt slate extraction or microwave assisted extraction also can be used here. This next is the astaxanthin, so which is obtained from the microbial species, that is algae, microalgae, Hematococcus velalis. So it is the compound organism which is used, microalgae, and it is a red carotenoid with powerful antioxidant and anti-inflammatory property. And we could able to find this one in case of many cosmetics also, this astaxanthin. It is also used in eye health, promoting eye health and joint health supplements, used in cosmetics like anti-aging and skincare products. It is also added in the food beverages and aqua feed, so which is helping us for coloring compounds nowadays, which we are finding the different colors of beverages like sodas and all. So that red color is due to the because of this astaxanthin compound. It can be extracted by means of solvent extraction methods like ethanol, methanol, or hexane is used. And apart from this, supercritical carbon dioxide extraction is the method used here. Then ultrasound assisted extraction and fermentation based production is also carried out, as it is an algae, and we can able to isolate by means of the fermentation is the best process which is used for this astaxanthin extraction. Silmarin, which is a well-known compound for hepatoprotection, which is obtained from milk thistle Silpum marianum. It is the plant which will be having this violet color flowers, and it is majorly used in the main compound, which is obtained from Silpum marianum, and detoxification it is carrying out. It is also useful as a nutraceutical, as a and supports the liver health in plants and liver stock. Like in the animals, also we can able to use this silmarin compound. The extraction method for silmarin is it is carried out by means of grounding, that is like cutting and making size reduction, and then purification will be carried out through the chromatography, and it can be extracted by methanol, ethanol, or acetone solvents. And is also carried out by supercritical carbon dioxide extraction and microwave assisted extraction is used. The other very good compound, which is called as ginseroids, this is called as the immunomodulatory and immunostimulant. Any type of diseases, any type of infections, this is the very best compound, ginseroids. So the main characters are it is a saponin with adaptogenic and neuroprotective and immunomodulatory effects. So it is obtained from the ginseng roots, that is the Panax ginseng. You can able to find this specificity of these roots are like the body of the root appears to be like a human body. You're able to find in the picture that the roots of this ginseng will be having the human-like structure. So indicates that they are having the immunostimulation property in the humans. The uses are used in the stress management and cognitive enhancement. It is also used in energy drinks and dietary supplements, and in some cases, like I think in Korea region and all, they'll be using this ginseroids as a normal vegetable. Ginseng roots are used as a normal vegetable. So at a particular climatic conditions, it will be stimulating the immune system. Especially in this winter season, this is best supplement. And these extraction methods for ginseroids are like normal hot water extraction, as like our green tea. We can also boil the roots and then it can be taken in the form of tea. And is also used ethanol extraction method, ultrasound assisted extraction, and fermentation process. All these methods are used for extracting the ginseroids from ginseng roots. And lastly, we just need to see what are the various factors which are affecting the bioactive compounds activity. One is the chemical diversity, biological activity, source of origin, based on the origin, like from where we are going to obtain the parts of the plant. Also, the compound quality will be varying. And using a right extraction method and right isolation process will be helping us to isolate the bioactive compounds in the proper manner and purified manner. Bioavailability is also need to be checked based on the purity of the compound only. We could able to find the bioavailability of the particular bioactive compound. Next is the safety and toxicity. So we need to check the before going for the formulation development. We need to check the safety and toxicity of the bioactive compound, and then we need to identify the synergistic effects, like in combination with the other drugs, how it is going to act, whether it is having the potentiating or antagonistic, agonistic, any type of effect we need to screen. And then we are going to check the antioxidant property, because all the type of bioactive compounds will be having the antioxidant property, as they are going to have the phenolic group. So the especially like we'll be finding the apart from the usage of these bioactive compounds, we'll be finding the some of the challenges, like complex extraction process. We'll be finding the sometimes the extraction may not be complete, and purification process also it is a tedious process, and limited availability and sustainability of natural sources. As we see nowadays, there is an endangerment of many of the species. We are not even able to know the whatever we have seen the common plants also. We are not able to identify the natural compounds. And potential toxicity and side effects at high doses. It doesn't mean that bioactive compounds are safe, but using in the large dose may lead to the toxicity also. And there are some of the regulatory barriers for the approval, like it cannot be done in the smooth way. So we could able to find all these challenges for this bioactive compounds extraction, and future directions, like This is like biotechnology approaches like genetic engineering to enhance the production. We can also use the genetic technology, gene technology. So thereby the production, the quantity of the bioactive compounds can be increased. Next is the in vitro synthesis using the microbial platforms and exploration using the deep sea and extreme habitats and personalized medicine with bioactive compounds based formulation. So we are going to make the preparation of the compounds which are natural and which are safe. And this is like by means of identifying the bioactive compounds, one can able to have the good technology and also we could able to obtain a good kind of product into the market. Now, that's what I wanted to say with this bioactive compounds. So thank you all the participants for listening patiently and also uh, for understanding the concept. Thank you. Sir, is there any questionnaire like?
Thank you, ma'am, for your wonderful presentation that enriched us with insights on bioactive compound from natural sources.
Thank you,
Dear
sir.
participants, if you have any questions, please drop them in a chat box. Our speaker will address your questions.
Orally they can ask. Orally they can ask. No?
Madam, one question from Mubasir Bhatt.
Yes, sir.
Could you please explain what makes green tea anti-inflammatory?
Green tea anti-inflammatory. Yes.
Yes, ma'am.
So green tea, that is the epigalactogenin, is the compound which is having the antioxidant property, and it is also considered as a flavonoid. So these two are the major compounds which are responsible for the anti-inflammatory property and antioxidant property of the green teas.
Is it possible?
Madam, one more question from Annegowda sir. It is possible
Yes,
to
sir.
have these informative slides.
Sir?
Is it possible?
It is
Didn't
possible to
understand.
have.
Is it possible to have?
Is it possible to have these informative slides?
Yes, sir. I'll share you, sure.
Uh, and one more question from Tinku Gupta. What are the modern isolation methods for the crude drugs?
Yes, sir. I've shared in the slide that there is a nowadays whatever the methods used in the earlier one is like oxidation and direct boiling, but nowadays we are having the microwave assisted extraction, ultrasound assisted extraction, then we are having the solvent extraction and supercritical fluid extraction. These are the modern methods which are used for extraction purpose. And also eco-friendly like green solvent extraction is also the modern method which is followed for getting the purified compounds.
Madam, another question. Which is the best method to isolate bioactive compounds?
So like sir, there are different factors are there based on the type of compound and its solubility. We need to choose the method because there are different methods are there. For example, I'll say azafoetida. Azafoetida is the one which is having a non-polar nature and it is a resin. So for this, we need to use a non-polar solvent. Then only we can able to get the complete extraction or complete isolation of the compound. But when you wanted to go for a coloring compound like carotenoid, in that case we need to choose the water and then we have to choose with the direct boiling or solvent extraction method can be used. So based on the type of compound, we need to choose the method of extraction. And based if you doesn't know that what type of compound it is, first we have to perform the like normal extraction with a water boiling, direct boiling, then separate that extract and carry out the test. If all these tests are not able to find out, a common general traditional method is soxylate extraction by means of using the ethanol. We can able to carry it when we are not knowing the compound type of compound.
Madam, next question. How to
Yes, sir.
fix the dose of active compound?
Yes, sir. So first thing is like after isolating the compound, we have to go for the acute toxicity studies. After performing the acute toxicity studies, we'll get to know that at which dose we are finding the safety and therapeutic action of the drug. And based on that, we are going to fix the safety dose of the bioactive constituent.
Madam, how do advanced techniques like LCMS and NMR improve the identification process?
Yes, sir. LCMS is the one where we can able to get to know that what type of compound is present in the particular extract. Because like instead of going with the column chromatography and carrying out all the time and using the lot of solvent, non-polar solvents, this LCMS method is the advanced technology nowadays which we are finding to determine that what type of compound is present in the extract. So based on the type of compound, we can able to find out that what kind of activity it will be having. So like for example, if it is a flavonoid, so we can able to find out that flavonoids will be having the anti-inflammatory, antioxidant property, and many of the chronic inflammatory diseases also. So based on that, we can able to find out that, and NMR is the one which is having the like majorly for identifying the compound. This number of carbon atoms and hydrogen atoms will get to know from the NMR studies. Just structure, approximate structure can be determined by the NMR studies.
Madam, on what basis plants are selected for isolation?
That's what I told in the slide. There is a different methods. One is based on the folklore usage. You can select the plant, or based on the traditional system. Like every geographical area will be having the one kind of plant will be considered as a auspicious or very important to the life. So those kind of plants can be selected, are based on the your taxonomical area, taxonomical characters which are available, we can able to select.
Madam, last question. Why
Yes.
the bioavailability of bioactives is low? How do we enhance the same?
Bioactive constituents bioavailability is low. In that case, we need to identify that which is the compound causing the absorption process hindrance. Like example, a glycoside compound is there. Glycoside will be having the two compounds. One is the glycon moiety and glycon moiety. If the glycon moiety is attached with the glycon moiety, then only we could able to find the 100% viability of the particular glycoside. If there is a detection or separation of the glycon and glycon moiety, then we cannot able to find the absorption of the glycon moiety. So in the glycoside, glycon is the one which is showing the therapeutic activity, and glycon is the one which is helpful for the absorption process. So we have to enhance the absorption by means of adding any of the molecule like which is a water soluble, because when it needs to enter into the systemic circulation, it should cross the cell membrane. The cell membrane is the one which is having the water soluble nature and as well as lipid soluble nature. So based on the compound, if the compound is having both water and lipid soluble nature, then it can able to enter into the systemic circulation. If not, then we have to alter the solubility of the compound by means of various salts process or like derivatization process.
Thank you,
Yes,
ma'am.
sir.
Now I
Thank
request our HOD sir to deliver the vote of thanks.
Thank you, Manoj. Before I say a note of thanks, first of all, I thank you, our speaker, that wonderfully he presented justified the topic. The slides were visually appealing. They are professional and they are well designed, and also very much informative. Keeping in view all the contents delivered by you, madam, there are still many more questions are there, and we have shortlisted and we have delivered, and we just selected questions we have put before you, and you have beautifully answered all those things and justified. I, on behalf of Adichunchanagiri University, Adichunchanagiri College, Sri Adichunchanagiri College of Pharmacy, I thank you for accepting this invitation and delivering the talk on the bioactive compounds and isolation and the purification. As we have started from the objectives to challenges, there are ranges of things you are delivered, and you tried at a level best to justify the contents, madam.
Thank you, sir.
So with that, I thank you for that. And it is also need of the hour that instead of as we are going for synthesizing the many molecules and failing to get a desired structure, but the bioactive molecules which are naturally present in the sources, if one could able to do it and isolate it, really it will be very beneficial for the institute as well as for the industry, madam.
Yes, sir.
Yeah, and I thank you, our principal, madam, Dr. Bharati, for creating an opportunity to go for this webinar. And I especially thanks to Dr. Nagprashant, who has taken very much initiative from the 1 week to design and everything. And I also participate all participants that wholeheartedly they have taken participation in this webinar, and I thank you all the HODs. So as per the report, it is saying that for the today's webinar, there are 650 participants have taken role in this webinar, madam. Thank you very much, madam.
Thank you, sir. Thank you so much for giving me the opportunity. Thank you, ma'am.
Yes.
Thank you.
Just a minute.
Thank you, Anjana madam,
Thank you,
for
sir.
accepting our invitation and making
Welcome,
this
sir.
webinar very successful.
Welcome.
Dear participants, the feedback link is there in the YouTube chat box. Please don't forget to fill that feedback link for getting your certificates dropped into your mailbox. Please give us a time of 1 or 2 days so that we can design your certificates and send to you mail. Thank you.
Thank you.
Thank you, ma'am. Ma'am, you
Thank
can
you, sir.
start leaving, madam. Thank you.
Thank you, sir.
Sure. Yes. So, after 2 weeks now, I'll ask that LCMS person to give a demo.
Done, sir. Same.
LCMS on ಇದರಲ್ಲಿ ಹೈದರಾಬಾದ್ ಇಂಡಸ್ಟ್ರಿಯಲ್ ಇದೆ. So this will be very much connected to that topic. This is over.
And a